Right. Shaka Prince is next. Prince is from Michigan State University, and he is a rising sophomore. Shaka, you ready?
Hi guys, my name is Shaka Prince, and I had the wonderful opportunity of working in Assistant Professor Catherine Prater's lab over the summer with another summer volunteer named Elena Cooley Rivera. We worked on analyzing microbial morphology in resilience to Alzheimer's disease. But before going into the research, I'd like to first review why Alzheimer's disease study is important in the first place. Over 7.2 million Americans are dealing with Alzheimer's disease as of 2025, and this number is projected to increase to about 14 million by 2050. This increase in people with Alzheimer's disease is due to the advancement in healthcare technology, causing people to live longer lives and as such be at a higher risk for Alzheimer's disease. This increase in people with Alzheimer's disease also means an increase in cost of care for them. As of just 2025, the estimated cost of people with Alzheimer's disease is 384 billion, and that's also projected to, of course, increase to about $1 trillion by 2050. Knowing and understanding these facts makes the research so much more crucial. And yep. Oh, sorry, I don't know how to do that. But what is Alzheimer's disease? Alzheimer's disease is defined as the accumulation in the brain of extracellular amyloid beta plaques and intrafibrillary hyperphosphorylated tau. This definition of Alzheimer's disease leads us to categorizing the brain in three when studying AD. The first being the resistant brain. Sorry, how am I saying? The first being the resistant, meaning the individuals developed no significant pathology. The second being the affected Alzheimer's disease diagnosed individuals. And the third being the picture right there is the resilient individuals, which is the group that we're more focused on. Resilience meaning that individuals developed significant pathology but showed no signs of cognitive dysfunction. Although plaques and tau are the hallmarkers of Alzheimer's disease and must be present for the diagnosis of Alzheimer's, it is also important to note that Alzheimer's disease patients also experience high levels of neuroinflammation. Knowing this has led us to further study the resident immune cells of the central nervous system named microglia. Microglia are key components of the inflammatory processes of the brain. Microglia are influential immune cells accredited with many important functions, just as shown in this image. It has come to be understood that microglial shape plays a huge role in the functions they serve. The more traditional definition of microglial is this sort of linear path with a ramified state, with the branches being called the ramified state, and then the amoeboid being the more active and phagocytic state of it. But with the advancement in the study of Alzheimer's disease and microglia, we've come to understand that the shapes they can take on in relation to their functions are much more complex, with the more modern understanding having this sort of like circular shape with many different shapes serving many different functions. So understanding the microglial morphology plays such a crucial role in the shape in the functions they serve, we were led to wonder, does microglial morphology differ in, oh, what is it then? Does microglial morphology differ in resilience to Alzheimer's disease? So for this study, we were able to successfully analyze the microglial morphology of 10 individuals, all split into the resistant, resilient, and affected individuals. I want to point out to the fact that the resilient individuals, right here in the middle, had both a high ADNC score and a high cognitive score, meaning that they had signs and significant pathology, but showed no cognitive dysfunction when we looked at them. But for the actual analysis portion of the brain, of the microglia, sorry, it first required an immunohistochemistry in which tissues were stained with cell-type markers. This is then followed by a thorough imaging process in which surfaces are created for the microglia in both layers 2, 3, and 5 of the prefrontal cortex for all the individuals. These surfaces and the following filament tracings were done in a software named MRS. And then these are the microglia colored in the cyan. After that, we then did filament tracings where we traced out all the microglia and they ended up kind of looking like this. An important aspect of the MARS to know is that it allowed for more accurate tracings with its three-dimensional perspective rather than the more common two-dimensional perspective that we're used to. Over the summer, Ellie and I were able to successfully trace over 300 microglia. After the tracings, we then analyzed the microglia through the show analysis method. The show analysis method is an analysis method in which circles are placed around the microglia with a distance of 5 microns, and the number of filament intersections are then measured out. After measuring the filament intersections, we then placed them onto a graph, placing the number of filament intersections on the y-axis and the radius on the x-axis. The data revealed a significant difference, with the resilient individuals, colored in blue, having the highest number of filament intersections, followed by the affected, and then lastly, the resistant individuals had the lowest number of filament intersections. These findings were kind of surprising because if we followed the linear path of microglia shapes, we would have expected the resistant individuals to have the highest number of filament intersections, followed by the resilient, and lastly, the affected individuals would have the lowest number of filament intersections because you'd expect them to be more amoebic. So for conclusions, understanding that seeing that the resilient individuals have the highest number of filament intersections shows to show that the microglia may help maintain cognition in the presence of pathology, because it shows that the resilient individuals have microglia doing something different, leading them to have longer branches. It also shows the microglial morphology is much more complex than the classic linear pathology, because the data with the classic linear pathology, we expected the resistant individuals to have the highest number of filament intersections. For future directions, I believe a larger cohort would help ascertain the findings that we found in this data. I also believe that due to the lack of diversity in Alzheimer's disease research, creating a more diverse cohort would allow us to better generalize the findings in this data. And then finally, I'd like to acknowledge everyone that helped this happen. Julie was so helpful when I got to Seattle. I'm from Michigan, so I didn't know anything about Seattle. And she's been very helpful with that. And then Dr. Ellen Bogan and everyone in the JDev lab. Of course, Katie, she saved me so many times. And then, yeah, the resources that were provided for us. Thank you. Thank you so much.